Okay, so this talk is sort of filling in for another speaker who had to move his talk, but in I guess a week in a couple days we won't have the typical lecture because there's another lecture that's coming from out, not for this course, but on campus that overlaps a little bit with this. So anyway, but either way, this is a talk that kind of probably overlaps a little bit with what you've been hearing in other contexts. For instance, with physiologic noise or with the physics of the signal and things like that. However, I thought I'd try to put this in sort of one context. Like when you're doing an fMRI experiment, what generally do you have to think about that might influence your MR signal or your fMRI signal? Everything from the physics to the physiology. And, of course, you want to keep all those things as constant as possible when you do your experiments so that you can, make a very subtle comparison and say it's real. You don't want someone to, for instance, let's say you're doing two populations. One population, drinks a lot of coffee. When another population doesn't, you might get a subtle difference if you average enough between the coffee drinkers and no coffee drinkers, even if that's not what you're looking for. So things like that. So I'm just going to start off with this diagram that I showed before. So, our goal is to say something about neural activation through this hemodynamic filter and also with our measured signal. So typically, the types of neural activation that exist are, well, there's probably many other types, but these are all things that could influence the bold signal in different ways. And in this talk, I'm actually not going to go into detail as to like maybe gamma frequencies cause increases in bold, beta frequencies are associated with bold decreases, and there's a whole literature on this, all these types of neural activation. But suffice it to say that, there's been some stabs at trying to, separate inhibition from excitation. Not perfect, not very useful. People have looked at frequencies, coherence, transients, even subthreshold potential people claim might affect bold a little bit. So with the measurements, and I'll get into this a little bit more in the next slide, but we can do a lot of different things. I mean, typically with fMRI, now the common thing is just to look at the magnitude of activation or to look at the connectivity, potentially within the brain. But there's so many other things to look at. You can look at latencies as they spread across the brain, correlations, fluctuations, transients, undershoots. All these are measures of signal that might have some meaningful information to pull out. And of course, with the hemodynamics, you're up against this spatial and temporal hemodynamic filter. And within every voxel, you get a different distribution of blood, either arteries or veins, or large vessels or small vessels, or some mixture. And that strongly influences the bold signal change in every single voxel. So you might get an average percent signal change of 5% here, but if you looked at maybe one voxel with a big vein, that might be 20%. And so you have to keep that in mind as well. And this especially has become relevant with looking at big areas of activation. Just mapping them is okay, but it's become more relevant in the context of doing layer fMRI, where there's this systematic bias of large vessels in the upper layers, and then it goes down into capillaries. So that's something we're still struggling to get rid of. And of course, the noise, we're still struggling to get rid of. There's thermal noise, which is just intrinsic to the scanner and your system, your RF coil. There's other things like motion, physiologic, respiration, cardiac that are all physiologic noise. And even though we have some rough models of what they are, we can't just model them and regress them out. It's not that simple. It's very nonlinear, and it's very spatially variant. So we can't just use a simple model to get rid of them. And typically, the temporal signal to noise is on the order of 100 to 1. Our functional contrast to noise can be as high as 10, but typically is almost never that high. It's about close to 1, actually. When you're doing activation, it's a 1% signal change against 1% noise, or maybe 5% signal change against 1% noise. That's maybe 5 to 1 at the most, but it's closer to 1. So Okay, so this is sort of to give you an idea. This is a movie of everything going on with a left and right finger tapping, and this is a hemodynamic response. So I'm not going to talk at all about this. I just figure I just put it up as an overview about what measures we actually can obtain from MRI and functional MRI. So as I mentioned, you can look at, and all these have been reported in papers. And so it's good to keep this in mind when you're designing your study of potential things to look at. You have location of activation. People have, in the early days, reported just simply the center of mass, extent, magnitude, width or shape, latency, or you could also have other measures of shape like, tortuosity and things like that. The latency of the hemodynamic response, or for the hemodynamic response itself, you could have the width of the hemodynamic response or the shape of it. Transients within the activation response, like the initial undershoot, the pre-undershoot, or the initial transients. Sometimes you have a transient at the end of an activation, post-undershoots. Changes in activation over time, repeats, task intensity. fMRI adaptation is all based on this. Multivaxal pattern, that's information-based fMRI. Connectivity, mutual information is another way of looking at that or describing it. Decodability, actually, I have a visiting scientist here in my lab who's doing decoding of fMRI signal with event-related responses. And so that's another thing you could actually look at, the decodability over time. Resting state, of course, dynamics of resting state, and also spatial temporal components, overall latency, pulsatility, information, entropy. You could also do things with the contrast. You could look at the flow to bold ratio to get metabolic rate. You can look at spin echo to gradient echo ratio to get vessel geometry. People have tried to do that. So there's all kinds of information that you can get that's not necessarily overlapping from the data sets, so that we haven't really, I would say, so I'm going to try to say a lot of controversial things, as this is the whole nature of this whole course. But I would say that most people in the literature have not really even tapped the potential information that's contained in the fMRI response. So the anatomy, of course, you can look at, typical anatomical scans are white matter, gray matter, CSF. You can look at myelination. That's a little bit more tricky, but we are able to look at myelination. Gyrification. You can even create measures of fractal dimension. That's interesting as well. I think that's even potentially something that AFNI can do. Fractional anisotropy with diffusion weighted. And you can have various measures of blood volume and iron deposited in the brain. And then you can also do things like use other advanced pulse sequences to do what's called magnetization transfer, spin locking to look at T1 row, and other things like that as well that we're still not quite sure what they represent physiologically. Magnetization transfer actually modulates the contrast with macromolecules. So it's kind of interesting to see if you can actually look at macromolecule deposits. Okay, so what I'm going to talk about today mostly, it's not a long talk, it's maybe about 30 slides, talking about what physical parameters and what physiologic parameters affect BOLD. So starting with the physical parameters, TR, TE, field strength, flip angle, diffusion weighting, magnetization transfer pulses, slice thickness, voxel dimension, and readout direction. So the reason why I'm trying to emphasize these again is that when you design your experiment, you have to, depending on what you're looking for, you have to kind of keep these in mind, that they might systematically affect your signal. So while it's important to keep everything as much the same as possible, if you do have to vary things, it's important to understand the way they vary, potentially. So this is a really old slide. This is actually taken from my PhD thesis when I was doing some basic work on looking at the effects of TR. And so what it is basically, this is the, so whenever you collect a fMRI time series, you start out, and I think what the scanner does now is remove this transient. That's the difference between the signal when you first excite up the first slice, and then the steady state over time. And it takes about 10 seconds to reach steady state. And so it turns out that when you're going really slow with a really short TR, that gives all kinds of time for the longitudinal magnetization to relax up. So the difference between the first one and steady state is almost nothing. And so here I measure the difference between the first image and the steady state. That's how much the image itself is saturated, how much the signal's pushed down by the fact that you're exciting it more and more rapidly. And this is a measure of the percent bold signal change. And I wouldn't say it's even, it's one subject and one experiment, and so this might have some other systematic differences. But the point I'm trying to say here is that as you go faster, so this is where I have a 90-degree flip angle, 6 seconds, 4 seconds, 3 seconds, 2 seconds, 1 second TR. As you go faster, up to 0.3 second TR, right here, the signal, the steady state signal gets lower and lower. So signal to noise goes down. But what also happens is that, at least this is looking at a few slices, that the bold signal change goes way up. It goes from 2% to almost 4%. So the reason for that I figured here are twofold. One is that you have less partial volume averaging from non-bold tissue. Like, for instance, when you have a long TR, you have all the CSF in the image. There's CSF everywhere because CSF has such a long T1. Now it's everywhere in the image. Partial volume averaging, it's like more tissue is there that's non-bold related that just causes maybe the percent signal change to go down. But what's interesting, though, this doesn't follow exactly this curve. It seems right around a 1 second TR. And so 1 second TR is actually really nice also because the images themselves look flat. With a 1 second TR and a flip angle of like around 90, the images are flat. So if the subject is moving a little bit, you don't get these edges that will show up as edge effects. It's all pretty much flat. However, at least in this particular case, there's more inflow effects. So fresh spins that are outside your plane. So now that your plane that you're imaging is saturated with RF, those spins with activation accelerate, they flow in. And that's causing the activation signal change to go up for a totally different reason than bold. It's all inflow effects, pure T1 effects. So this is actually capturing somewhat the enhancement of inflow as you go shorter and shorter TR. This is not a big deal when you do whole brain multi-slice imaging because of the fact that it might only affect the bottom and top slices typically, but it's something to at least be aware of. So this is actually when I start reducing the flip angle. So you have less and less inflow effects, but not a total reduction. And you still have an elevated bold that's a little bit higher than typical. So anyway, that's the effect of TR. I'll get a little bit more into flip angle as well. Okay, so shorter TR increases samples per unit time, which is actually a really good thing for estimating the noise. Increases inflow effects on outer slices, so that might artificially enhance your signal a little bit. Definitely decreases the signal to noise because the overall signal is going down as you decrease your TR. But it also allows physiologic fluctuations to be characterized and removed. The faster you sample, as Bob Cox was saying in the last lecture, the better you are at if the noise is not, if it's not temporally correlated, you can actually characterize it. Like you can now characterize cardiac frequencies and respiratory frequencies as peaks and then get rid of them. So that's really nice in that regard. It also just gives you more samples. So it's always, even though your signal to noise goes down, it's always better from a statistical point of view to get more samples. That counteracts the signal to noise going down. Okay, effective TE. So this is also one oldie but goodie slide. This is a simultaneous sequence in which I collected, with every TR, I stepped the TE and then cycled that for a really long experiment during rest and during activation. And you don't have to look at any of these numbers, but just know that this is the gradient echo signal, the raw signal as it decays with echo time. Well, at least it's log transformed, so it's like an exponential. And this is the spin echo signal as it's decreases with echo time. And this is during rest, this is during activation. You don't see much difference. And here is basically this same data now showing the echo time dependence with gradient echo. So as you lengthen your echo time, the percent signal change goes up. As you lengthen your echo time here, the percent signal change goes up, but not quite as much as with gradient echo. And that might look like spin echo is about the same here in terms of percent signal changes, but what really matters is the signal difference. And so this is the same calculation, but instead of a percent change, we're looking at signal difference. You can report things in percent change, but the real contrast is determined by this. So this shows that gradient echo, the signal difference is much higher than with spin echo. So much higher contrast. And also it peaks at the echo time equals of about 40 millisecond, which happened to be about the T2 star of the baseline. And this is actually happens to be about the T2 of the baseline right here, about 100. So that's echo time dependence. So and this is another thing too that is important to note. Some people say, oh, well, we have to pick just the right echo time or else otherwise we won't get any bold. Well, in fact, this is a pretty shallow curve. You can actually have an echo time anywhere from 15 to 80 and still get, or even up to 120 and still get about the same contrast. So you have a wide range of what echo time you pick if you want to. Generally, I like to pick lower echo times because then you don't get susceptibility dropout. So I usually try to pick almost the shortest echo time I can. It doesn't really matter that optimizing that contrast to noise curve. And actually, what I should do is plot this against how not the difference, but the difference per signal over divided by the temporal noise. And if you do that, I think this will even be much flatter too, because the noise also shows an echo time dependence to some degree, breathing at least. All right, field strength. I was going to give a whole talk about this, and I still might, but this is basically, and you don't really have to look at these curves. This is just showing the contrast as a function of echo time, and I already showed this as well in a previous talk. Echo time with various different baseline T2 stars. So if you're skating at 3 Tesla, your shim is really bad so that your signal happens to have a T2 star of 15 milliseconds, you won't ever get much contrast. You'll have less contrast than even at 1.5 Tesla if the shim is good. So it's important to have a very, very good shim at 3 Tesla. And also, and that just shows you the summary of that right here. But so basically signal noise goes up, the percent signal change goes up due to bold effects just being additive at higher field strengths. Physiologic noise tends to go up as well. Even though contrasted noise overall goes up a little bit, it also depends on the physiologic noise, how much that dominates. And I'll talk a little bit about that later. But off-resonant artifacts, if something has a poor shim, it shows up much more at higher field strengths. And then RF power deposition goes up. So if you're doing like certain sequences called like fast spin echo, where you apply a lot of RF pulses that are like 180-degree pulses, you have to start worrying about, well, you don't have to worry, the scanner's worrying for you, where it will stop if the RF power deposition is too high. So, okay. And this is just one quick word on noise because it will be relevant to the next part. That is the difference between physiologic noise and thermal noise. So if you have a phantom in the scanner, all the temporal, this is basically temporal signal to noise versus overall signal to noise as measured by within one image. And typically this should be a line of unity. These should be all equal. And in a phantom, they're pretty close. So the temporal signal to noise is equal to the thermal, to the spatial signal to noise. But if you put a living brain in the scanner, this goes up for a while, and then it peaks out in gray matter at about 100, a signal to noise of about 100 to 1. And that's important because if you're scanning and physiologic noise dominates at 7T versus 3T, you're not really going to get any higher signal to noise, temporal signal to noise than 100 to 1 because physiologic noise is dominating. So you really only reap the benefits of high field when you start to image at resolutions such that your signal to noise goes down, such that it's more evenly dominated by thermal versus physiologic noise. So this is where you get the real advantages. This, like right about there, is about 2 millimeters at a 2 millimeter resolution at 3 Tesla. That would be about a 1 millimeter resolution at 7 Tesla. So that elbow of that curve where you kind of want to be, you don't want to be wasting resolution because you're dominated by physiologic noise, is different at different resolutions for higher field. So higher field, it's smaller resolutions. Okay. So one thing actually is an effective flip angle. So whenever you do your studies, they say, oh, what flip angle do you want? And you can look it up in a book. There's what's called the Ernst angle. So the Ernst angle is basically given the tissue T1, you can figure out what the optimal flip angle is from a purely signal to noise standpoint, given your TR. And that usually this is the simulations of the signal to noise right here. And this is actual signal to noise, what the optimal flip angle is in the actual image. But this actually turns out to be what the temporal signal to noise is as a function of flip angle. And just like with echo time, and this is even more so, in gray matter, you can work at a flip angle anywhere from 7 degrees up to 180 degrees or 70 degrees and still have the same temporal signal to noise. So flip angle doesn't really make that much difference at all in your study. You can, in certain areas where if you're able to get rid of all your physiologic noise, and someday we will, then it will make a difference. But since physiologic noise dominates, flip angle, everyone's sort of careful to have like their Ernst angle. No, it makes almost no difference. So, and here's an example of an actual study. This was one by Javier Gonzalez-Castillo a few years back, where he looked at the bold response in raw units versus bold response in terms of percent signal change. And this is actually contrast to noise across flip angle, and everything's pretty flat right here. And this percent signal change is pretty flat as well. So that's basically saying you don't have to worry about flip angle. However, it does affect the image contrast a lot. So here's a bunch of image contrasts with a flip angle from 9 degrees all the way up to 120 degrees. And this is how the image looks right here. And you can see at 9 degrees, you hardly ever flip the spins down, so they relax back to longitudinal magnetization really easily, really quickly. So you get a lot of CSF. You get a lot of CSF in the ventricles. And this sometimes is a good thing because then you can use that. Your registration programs work better when there's contrast in the image. Like if you put in IMREG, it will work better with an image that's contrasty like this versus not. However, you get probably a little bit lower percent signal change in bold because you have partial volume averaging of CSF. You might get a little bit more reduction in somewhat of the temporal signal to noise because CSF sloshes around. You also will have, if you don't do IMREG, even if you do, you have edge effects if you move, much more so than if you're at a flip angle like around here where the image is flatter. So less edge effects maybe at this flip angle, but better maybe registration effects at some sort of flip angle of 30 or something. So that's at least something to think about. All right, diffusion weighting. All diffusion weighting this is just a simple pulse sequence diagram, and we talked about this at length before, but apply a 90-degree RF pulse, you apply a big gradient, and then you apply a 180-degree pulse, you can apply the same polarity gradient, and then image. So these two gradients basically massively dephase the spins. And then this gradient massively rephases them. And anything that's moved a little bit, so it moves through space, will experience a different gradient when this one happens. So that's why this is sensitive to diffusion. So if you move outside of your where you were first massively dephased, then you won't be rephased perfectly then after that. And the same thing can apply with this is a spin echo where you have an 180-degree pulse that flips the spins. With gradient echo, you don't have that luxury, so you have to apply a big bipolar gradient right here that just massively dephases, massively rephases, and anything that's moving gets rephased a little bit less. All right, so that's very good at getting rid of intravascular spins that flow a lot. And this is just a proof that it actually works. This is bold signal change, percent signal change. This happens to be at, I think, only at 1.5 Tesla. And you see with zero diffusion weighting, it's a 3% signal change. With a B factor of like 300, which is still kind of small, it goes down to 1%. So this just shows that about 2/3 of the signal at 1.5 Tesla is all intravascular. It's all big vessel effects. This is only about 1/3 at 3 Tesla and even less at 7, but still there. Okay, this is a little bit esoteric, but I like to mention it. This is called magnetization transfer, just so you know what it means. It turns out that when you have water processing in the magnet, there's a very sharp range of frequencies in which it resonates at. But it turns out that macromolecules are also there, and they're also surrounded by water, and they have resonant frequencies close to water. It's centered around water. However, because of these large molecules, they process at a wider range of frequencies. They have a wider range of resonant frequencies. And so you can give an empty pulse completely off resonance from the water that will be transferred, the energy will be transferred through the macromolecules and reduce the water signal a little bit. So you could have these really nice contrasts of macromolecules in the brain with MT pulses. Does it affect bold? Yes, it does. So unlike with diffusion that gets rid of all the free-flowing water-like stuff, like the intravascular signal, this is exactly the opposite. This gets rid of all the big molecules that are even having a hard time processing at the persistent frequency. And if you do that, it turns out you can actually enhance bold a little bit because you're getting rid of all that other stuff that's around it. It seems that the bold signal change, there's only a few papers on this. With the efficiency of the magnetization transfer pulse, the bold signal goes up just a little bit. And like I said, the reason for that is that it reduces some of the, extravascular, non-bold contributing signal. So it's sort of esoteric, but it's something worth knowing about. Okay, now looking at the effect of slice thickness. So it's very similar to resolution in many ways, but or voxels I mentioned, but this is basically the same volume looked at, but here's with 1 slice versus 2 slices versus 4 slices and so on. And it turns out that if you chop them up into finer and finer slices, the overall signal will go down because there's less volume in that voxel. But the percent signal change, so number of slices used, average percent signal change consistently goes up. And that, once again, this is a whole partial volume concept. You have less non-bold related signal that's contributing in each voxel. So then you have a larger overall percent signal change. A lot of voxels that are no longer activated, but you have a larger overall percent signal change. So here's just an example across the slices of an average signal change up to 5, whereas here if you just have 1 slice of the whole brain, it's only 2. So same things happens with in-plane resolution. And these are all pretty straightforward concepts, but it's important that they're real effects when we're doing fMRI. So this is awful reproduction of the images. This is a paper that we wrote back in 1998, basically changing the resolution from 2.5 millimeters squared to 0.62 millimeters. And this is looking at motor cortex activation. And this is a histogram of the number of pixels versus percent signal change. And at 64 by 64, which is the 2.5 millimeter resolution, you have most of them all centered around 4 or 5%. But it turns out that as you go to higher resolution, you have less around this average, but a lot more outliers. So those are these effects where you suddenly make your voxel so small that it completely is contained, it's completely filled by a vein. And so that will cause these 20% signal changes and so on. And you start getting that even at about a resolution of about 1.25 right here, but not that much. It starts really happening at about 1 or 2 or 1 millimeter here, or 0.83 millimeters here. So it doesn't happen until things get really small, but it's sort of a continuum though. Okay, this is just some basic MR physics, but I'm not going to go into any physics whatsoever. It's just all rules of thumb. So when you're doing your scan, right now, this is just important to know that when you're doing your scan, there's different directions. There's readout directions that you see, AP, SI, whatever. And that will make a big difference in terms of where the artifacts appear. So if you have off-resonance effects, then if you have readout direction that is, let's see here, where's my pointer? There we go. That this is echoplanar readout, and let's say you're basically, with echoplanar, you're sampling really fast left to right, but you're sampling really slowly in the y direction. And because of that slow sampling, any off-resonance effects will have huge effects in the vertical direction. So you have, so here's with blips up going in this direction, you have the front of your brain is sort of pulled out. If you blips down, the front is smushed down. And there are ways of correcting for this that exist, probably even within AFNI, but just to be aware that which way your subject's brains are smushed in the image is very much a function of what the readout direction is. And it turns out also the readout direction makes a difference. All images, no matter how good their reconstruction, always have some amount of Nyquist ghosting. And that will kind of appear subthreshold. This is sort of a long one, but a large one. And that depends on which direction your readout is as well. So basically, try to keep your readout directions consistent, because it will make a difference in comparing in the signal to noise and everything like that. So because there always will be some Nyquist ghosts that are looming below threshold. Also, it makes a difference in terms of how artifacts appear. So if your readout direction, let's say it's left-right, it goes this way. If you have like a pulsatile, like the sagittal sinus is often very pulsatile, that will look like a bunch of junk, kind of like through the image in the vertical direction. It will just be one direction. And with spiral scanning, it's a little bit different. With spiral, any off-resonance effects won't be manifest as warping in one direction. It'll be like blurring. So if you ever do spiral imaging, single shot spiral, it will be blurred to the amount that you have off-resonance effects. And then secondly, this is the thing I hate most about spiral scanning. I mean, I like spiral scanning in general, but if you're doing any visual cortex activation, at least from my experience, that the sagittal sinus, instead of pulsating with a nice, neat line that you can kind of look through, this actually has basically circular ripples that ripple out into the image in different ways for every single image. And so when you have a final correlation map, it will show the outlines of these ripples. So that's why I avoid spiral, because I don't like the ripples around pulsatile effects. Otherwise, it's a pretty efficient use of gradients. Okay, last part of my talk. What physiologic parameters can influence the fMRI signal? So off the top of my head, there's probably many more, but there's, the things that come to mind are hematocrit. Of course, the amount of iron in your blood will affect the bold signal, but how much? So blood pressure, blood volume in each voxel, as I sort of harped on as well already, and neurovascular responsivity and drug effects. So hematocrit, I mean, I've actually looked, and I only found a couple of papers exploring hematocrit effects on bold. And one I didn't like because they did the wrong way. They actually changed people's hematocrit by doing hemodilution. So it didn't really change the amount of red blood cells that were in the blood. It just changed your hematocrit by adding more plasma, which shouldn't affect bold that much. But this is more interesting. This is actually different subjects with different hematocrits were plotted at their average percent signal bold, which is really hard to get a measure of, versus hematocrit. And you see a small, it's nothing surprising, but you see a small relationship between hematocrit. People who are really iron deficient might have a 1% less signal change than people who, have extremely high hematocrit, close to 50. So anyway, hematocrit makes a little difference, not that much of a difference. It's hard to see these, but I've often wanted to study blood pressure effects, and it would be nice to have exactly like the hematocrit study here, where each dot is a subject. There's no studies that I could find where they just had a bunch of different subjects, different blood pressures, and just looked at their bold effect. Here was an animal study where they put in a modulator of blood pressure, either to increase it or decrease it. So here, basically, they injected this bolus of whatever increases the blood pressure, and it went through like this, and it was still hanging around after 300 seconds. So, and then what you see here then is it has basically two stimulation periods. This is stimulation only. This is with the blood pressure increase. You have not much of a change initially, but then you have a dampening of the bold effect once the blood pressure change actually kicks in. And that seems to, so the bold effect seems to be somewhat dampened. And I don't know exactly, I can't imagine the mechanism might be, there might be vasoconstriction going on that might change how much things are modulated. It's hard to say. With blood pressure decreases, a massive decrease in blood pressure, the bold signal is slightly dampened here, and it shows more transients. It shows more of an undershoot. Probably my favorite study in which was looking at blood pressure changes was one by Sanji Kim's group back in 2006. And there he, once again, was looking at rats, and he basically had sodium nitroprusside, which he injected to look at with activation. This is a stimulus is in blue right here. So this is before, during, and after. So this is sodium nitroprusside basically decreases your blood pressure massively. So if your blood pressure goes down way too much, then there's an opposite sign effect with bold. Since the flow response doesn't kick in, the blood volume response doesn't kick in, then basically all that's happening is you're measuring the amount of oxygen extracted from the blood with increased metabolism. So that's what happens there. If your blood pressure gets too low. They weren't able to induce like a pre-undershoot, but they induced a negative signal change. And that just shows this what the ranges are. So normal blood pressure is 128. This low blood pressure is like half of that, 64. So it's a really low blood pressure, but there's everything in between. So you can imagine people with maybe generally higher blood pressure might have higher signal changes on average, and then people with slightly lower blood pressure might have slightly more transients and maybe lower bold signal changes. Who knows? All right. Okay. But then even things like, so much less, I mean, this is one of the biggest, seems like one of the biggest problems in fMRI right now is dealing with respiration. Of course, you can have people breathe at a certain rate, which helps you filter out the signal. But if you're in a scanner and you take one big deep breath, if you're in a scanner and you just go, then your signal will show this response. Your brain signal will show globally this response right here that will last up to 40 seconds. It'll be this massive increase, well, not massive, but 1% increase, 1% decrease, and then it's basically looks like a bold event-related response, but it has a little bit delayed onset and a much longer post-undershoot. So turns out this Rasmus Byrne did this study where he had subjects breathing at certain timings, convolved this response with the timing, and actually predicted most of the signal in the brain. And what's interesting is that the areas that fluctuated the most, of course, it basically indicates a global flow change. And so a global flow change, if you happen to be over a voxel that has 100% venous blood versus a voxel that only has 1% venous blood, 100% venous blood will show much more of a signal change, given us the same flow change. And so that's the way of mapping out venous blood volume. So using this, he was able to map out really nicely venous blood volume because it potentiates bold in this way. And this was done a long time ago, too. This was an old study of mine in which I had subjects breathing 12% oxygen, which is less than room air, and then 5% CO2, causing a global vasodilation. And this is with brain activation right here. And you can see that with 5% CO2, the signal goes way up. With 12%, it goes down. If you superimpose activation on that, it gets blunted as you breathe 5% CO2, hypercapnia. Doesn't really get blunted as much with oxygen changes, but the oxygen level does go down a lot. And this is basically the relationship between the percent signal change with finger movement, not during these studies, but just with breathing room air, and the percent signal change with either any stress, either CO2 or oxygen. And you can see that these hotspots are all big vessel effects that show up high for activation and show up high for the stresses. So this is a way of getting rid of these big vessel effects too. Okay, nearing in towards the end here. The other thing that people have to worry about, especially when they do age studies, studies of different people with different ages, you might say, well, let's compare connectivity between younger versus older people. And oh, look, connectivity is less in older people. And the reason for that could be that, like this paper might suggest, that that when people get older, their neurovascular reactivity becomes less. So here's basically a study showing a Stroop task. And one might argue, oh, look, BOLD is less with the Stroop task for the older people a little bit. Therefore, their neuroactivity is a little bit less. But can you interpret that? Well, here's essentially what hypercapnia does. It causes a global signal increase, as I showed before, but it shows less of a global signal increase systematically with older subjects. And flow kind of remains almost the same. Flow is a little bit less too. So what this is indicating is that every single measure, and I don't know if, to the extent that hypercapnia is the same as neural activity, that could be argued, but every single measure of something like connectivity or the magnitude of activation between younger and older subjects is somewhat biased by this age effect. And you might say, oh, this is very small, but if you are really looking for subtle effects, this could be a major confound. So it's something at least to keep in mind. This is also neurovascular responsivity as it varies with people who have had a stroke, for instance, if you're trying to map out functional activation. So it turns out that with controls, you have BOLD, CSF, blood volume changes that are all occurring nice and behaved. With a patient where you look at the signal over the area similar to in the location of the area that had a stroke, the subjects are still able to do the task. However, and there's a CBF change, there's a flow change, there's somewhat of a volume change here, but BOLD is completely flat. So there's so something is screwed up in the neurovascular coupling with stroke patients. This is why this is, in general, it's hard to apply fMRI, especially BOLD, clinically, at least looking at stroke recovery, because you don't know whether that is a recovery of the neurovascular response or a recovery of neuronal activity in that regard. So that's tricky. It's a little hard to think about. And it's still not clear what the best thing to do is. So drug effects. So a lot of people want to do, and I just simply copied this from a nice review article. I forgot to put the reference, but I was about to go into drug effects, but I thought this sort of captures everything just perfectly. So basically, and I won't read this whole thing, but essentially, the points being made here are that so with drug effects, you could assume that regional changes are associated with modified neural activity mediated by intact neurovascular coupling, or they might be associated with non-neurally induced metabolic activity, such as developed from local drug binding. So when you inject a drug, you might have a global effect on the hemodynamics or effect on just the binding. You might have global changes on baseline flow, or you might have changes in terms of the heart rate, blood pressure, breathing that all affect BOLD as well. So when you're drug studies with BOLD are really, really hard. I remember brainstorming when I was collaborating a long time ago with Elliot Stein about how do you actually look at effects of nicotine when nicotine affects the neurovascular system so much. Can you actually tease these apart? Caffeine as well. Caffeine, and actually I'll get right into it right here. Caffeine. So caffeine actually causes a slight shortening of the latency of the hemodynamic response, and it causes a larger CBF change, but a smaller CBF difference, which is still a little bit confusing. Basically, when the blood, so basically it can be explained potentially by this study that shows 3 types of users, no caffeine, with caffeine. You can see there's a global constriction. There's a flow decrease, and so that might explain some of the modulation on bold and whatever. With a flow decrease, to maintain the same overall flow rate, velocities have to increase. So maybe that's why there's a faster arrival time here for bold. So anyway, so that's caffeine effects. So now, just to summarize, once again, we didn't really touch on everything. There's a lot of other things that affect bold, but I think that the key thing to keep in mind is simply that you have to be aware of the question that you're asking, and be ready for any systematic changes across your populations, or even I could have put in here time of day as well. So there's studies that claim that time of day affects the magnitude of bold changes. That might be due to changes in arousal or things like that. So people could drive themselves crazy worrying about every single thing. So I think the best thing to do is just do your best to try to keep things as controlled as possible, but not to worry too much about, just to after doing your best about keeping all the physical parameters and the physiologic parameters controlled, then just report that and understand that, yes, there can be many other variables. But another thing too is, let's say if I were looking at the caffeine study and I was just caring about latency, saying, oh, well, people who have caffeine are, their neural activity is faster, therefore it drives it harder. That would be probably an overinterpretation of these results given what the potential hemodynamic effects in the caffeine are. So, but if you just say, oh, look, looks like the caffeine doesn't increase the magnitude of the task, so you can probably do comparisons of magnitudes on neural activity with this. So anyway, there's all kinds of measures you can decide to do depending on what's most sensitive to these parameters. So with that, I'd just like to end it. Thank you. So any discussion today or nothing? All right. Okay, well, that's fine. I think that, so next week, I'll have another sort of brief talk on, next week, Tuesday, will just be on clinical, on why isn't fMRI more clinically relevant? And I'll go through the discussion with that. So, all right, thanks. Okay.